I hope you took some time to reflect on the Watson-Crick structure of DNA. Let's review the bonds of the DNA helix before we talk about the implications of their structure. Each of the strands is held together by covalent linkages. On either side of the sugars in the backbone is a phosphate that links the sugars together. These are very strong bonds. The two strands are then held together by non-covalent linkages. That is, the hydrogen bonds between the bases and the apolar stacking interactions provide the energy that holds the two strands in the double helical form. It's possible to interfere with these non-covalent interactions and to separate the strands of DNA so that you now have individual strands not in the double helix. This process is called denaturation, to make the DNA structure non-native. They denature from a helical structure to linear polymers of nucleotides, as indicated in the image. Heat or chemicals can be used to interfere with the hydrogen bonds and separate the strands. But when you reverse the conditions, lower the temperature, remove the chemical, the two complementary strands will reassociate, and this process is called renaturation. And so you go back to the double strand native form. The temperature at which the strands will separate or come back together reflects how well the base pairs are matched along the sequence, and it's also a reflection of the content of GC pairs. That temperature is called the melting temperature or midpoint temperature, Tm, and it's a measure of the stability of the DNA. So think about why an increased GC content would increase Tm. And it's because there are 3 hydrogen bonds for GC base pairs and only 2 for AT base pairs. So GC pairs render the molecule more stable, and so the higher the GC content, the higher the melting temperature. Not only can you denature and renature a DNA that was originally a single helix, but you can denature and mix DNA from different sources. That process is called hybridization. Hybridization can be used to compare DNA sequences. Let's say you take DNA from 2 organisms and separate the strands. You denature the DNA. Then you mix and allow strands to renature. Some strands from one species will renature with strands from another, creating a hybrid DNA molecule. The stability of that molecule indicates how related the sequences are. That is, how many of the base pairs in the hybrid line up and form hydrogen bonds. The X's in the figure indicate where hydrogen bonds didn't form. The more closely related 2 species, frequently the more similar the 2 sequences. So they have lower mispairing and higher Tm. This is a common way that was historically used to determine the similarity between 2 sequences. Today, we just sequence both DNAs. But hybridization is still important because it's used in methods involving DNA. So if you have significant sequence similarity, you can get hybridization. That level of similarity is often present for genes that make proteins that are essential across multiple species. We can take single-stranded DNA for a particular gene, and we can probe a mixture of single-stranded DNA molecules isolated from an organism. If we do this at high temperature, we select for stringent base pairing. That is, the higher the temperature, the more base pairs would need to form for stability. But at lower temperatures, you can form double-stranded DNA with mispaired bases, or they don't form base pairs at every position along the sequence. This allows us to compare sequences. Hybridization then reflects the similarity of the sequences. And this ability to use temperature to compare the hybridization has multiple modern applications. We're going to go through one of those that allows detection of specific sequences. Also, the ability to synthesize DNA was a transformational advance. Because we can synthesize specific sequences, we can sometimes know exactly what sequence is in a DNA sample. You can design oligonucleotides made by chemical synthesis, offering a whole variety of opportunities, including the one that we're going to talk about now. You can make short DNA segments that are labeled with a fluorophore, so it has fluorescence that you can detect, and you can probe for that sequence in multiple DNAs by allowing the labeled DNA to hybridize with the DNA that you're testing. When you do that, you form a hybrid that is marked by the fluorophore. Putting this together, we can describe a simple overview of the process for microarrays. There are more complex applications of this, of course. But we start with short DNA sequences that are synthesized and use robotic printing to place them in an array where we know what the sequences are at each position in the array. We now take DNA from two different sources. We can label those DNAs with a fluorescent base at its end, either Cy3 or Cy5 dCTP. Cy3 and Cy5 are fluorophores that have different absorbance and emission of fluorescence, so you can distinguish them. With this labeled DNA, we hybridize with the probe. We'll wash away unbound DNA, so the only fluorophore that remains is attached to DNA that is now hybridized to a sequence on the array. We use lasers of two different wavelengths, where each one lights up only one of the fluorophores. And we combine the images produced to see which sequences of DNA printed in the array hybridized to the DNA samples. And this tells us which sequences were present in those DNA samples. Microarrays detect defined sequences based on patterns of hybridization. You can use short segments of DNA or oligonucleotides that are immobilized in a defined pattern on a support material such as a 96-well plate, slide, or chip. The same array arrangement can be used to probe multiple DNA sequences, and this method allows you to detect sequences both visually and spectrophotometrically. Some arrays are very complex, and they can test many sequences at once. You can see from this array that's highlighted from the complex array. It shows the individual spots from a larger array where thousands of sequences are contained. The data are analyzed to determine which samples in the array are able to form the hybrid at each position of the array. Complex computational analysis can allow taking huge volumes of this data and interpreting it. So hybridization is the key in these microarrays. Why do you think microarrays are such a popular method for examining DNA? And with some modification, they're also useful for examining RNA. What type of experiments might this method allow that would otherwise be difficult?